Okay, so last time I talked about what a gene was. So last time I said a gene is a sequence of DNA that encodes a particular product, like a protein or some sort of a RNA molecule. But so, my apologies, a sequence of DNA. So basically, what is this DNA here? And so DNA is, the structure of it is fairly simple. You would have a phosphate group, so all DNA has a simple arrangement of the structure, where you have the phosphate group, you have the ribose sugar, and then you have some sort of a base. So these bases can include either adenine, guanine, and then you can have the pyrimidines, which are thymine and cytosine. So these are considered pyrimidines. And these are considered purines. So all DNA is just a collection of these molecules combined together to form DNA. And DNA is basically the library that contains all the genes, and is the library that contains all the proteins that you can possibly make, which if you think about it, is essentially, it makes up the central dogma of biology. Which you have DNA to RNA to protein. So basically, this DNA is responsible for you to make RNA and protein, and that will be used to function, for you to properly function for your cell. So this is just like a brief introduction to DNA, and we're not going to go over how DNA undergoes replication and stuff like that. We're just going to just give a simple introduction to how, just like the structure of DNA. And a famous study in the 1950s by Rosalind Franklin, what they did was they took a fiber of DNA, and they had an X-ray machine right here, and this X-ray machine shot through the fiber, and it scattered around onto a piece of paper to form that image right there. So this image tells us that DNA is composed in a double helix. But how is that? And usually when people look at this image, they find it very confusing. But the way you can think about this is these right here, these bundles here are considered nucleotides, and the backbone as well. So we can label these as red, and we can label this one as blue. So what basically ends up happening is that if you trace it, actually I should use the red color there. So if you trace this right here, it essentially looks something like this over here, where that where that backbone crosses the blue backbone right there, and it tells you that this image tells you that the backbone of the DNA is helical in shape. So DNA backbone is helical. And because there's two strands that are connected to one another, it's considered a double helix. So what is happening here is that your adenines bind to your thymines, and your guanines bind to cytosines. So the way how this particularly works and why guanines bind to cytosines is because the shape of a guanine has 3 possible locations where you can have hydrogen bonding, and so is cytosine as well. So guanine and cytosine are going to have 3 hydrogen bonds to one another, whereas adenine and thymine, they only have 2. So what is happening is that when you have one strand of DNA, so let's say this is adenine. At the same time, you're also going to have a thymine bind to it. In this kind of a fashion. So because A's and T's are binding to one another, and G's and C's are binding to one another, that essentially explains complementarity. And then the reason why I drew it like this is the way how typically these DNA molecules will bind together. And these carbons are written down as 5' here and a 3' carbon here. So essentially what is happening here is that DNA is anti-parallel to one another. DNA is anti-parallel. So this basically means that one strand in the double helix has a different complementarity, has a different polarity, my apologies, to the adjacent nucleotide. So basically this is how it looks like, just the simple structure of DNA. So hopefully that helps.